together to be scared. And I don't handle it. It's like 20 pounds. Yeah, they always, right, a ton of life. So that.
Okay. Nice to be back. So a reminder that we will have class on Thursday here, but the next Tuesday we will not have a class here because, as I said from the beginning, the Tuesday before Thanksgiving, I always record that lecture and post on D2L. So catch up to it there, those of you that need to travel. Questions about the course so far?
Fine.
Or about the material we talked about last time? Good, yeah.
Was the final exam number efficacious? Like
the final exam is comprehensive. We will try to pull some from all throughout the course, but it absolutely emphasizes chapters 8, 9, 8 on activity dependent plasticity, 9 on experience dependent, and 10 on social, and the epilogue.
Thank you.
Good. You know roughly how much it's emphasizing? Maybe like 50% of that.
I don't. It's more than 50%. Okay, for sure. Okay, but I can't tell if it's 63.7 or 63.8. But yeah. Okay.
Yeah.
So now we're gonna. I don't know why I put this here, but we'll get back to it. Now we're gonna talk about Molyneaux's problem. You remember that, right? Where the problem they wrote to Locke about, that Locke published back in the 1600s. What would happen if a man had been blind all his life and then as an adult been made to see? Would he be able to tell which was a cube and which was a sphere? And as Locke points out, and as Molyneaux pointed out, usually when you put this question to people, because the person knows perfectly well what cubes and spheres feel like, everyone's first guess is, of course he'll know which is the sphere and which is the cube. But then when you make them think about it some more, they think, no, he'd have to learn what those protuberant edges look like to his eye. And in all of it, there was the assumption that he, after that, would be able to tell a sphere from a cube. And it turns out that Locke's thought problem turned out to be a real life problem when in the 1700s, people realized that sometimes when people are born with cataracts, that is lenses that for one reason or another are very cloudy, such that light gets into their eye, but they can't distinguish any form vision. In every practical sense, except for telling you whether it's day or night, they are blind. And people realized that if you did surgery to destroy the lens and pull it out, that you'd get this cloud out from in front of the vision, and they wouldn't be able to see. Now, those of you who have glasses to know that things wouldn't be in focus, right? On the other hand, I'll remind you of visual science. Most of the refraction that happens to focus the image of the outside world onto your retina, most of that is done by the cornea, the curvature of the cornea, which is fixed. And so these individuals, if glasses had been available, they'd need glasses, but they'd be able to see for the first time in their life. And the pioneering British surgeon who did this, a guy named William Cheselden, talked about a case that seemed to indicate that Molyneaux's problem was exactly as Locke and Molyneaux had predicted. So he removes the cataracts from a boy who was a teenager at the time. And sure enough, when he first shows him cubes and spheres, the boy can't tell which is which. But according to Cheselden, he eventually was able to make use of his vision, just like, Locke and Molyneaux had predicted. And Cheselden was totally wrong. And so, in fact, if we read his report, it's English. You can understand it. We see that his interpretation that the boy eventually learned how to make sense of vision, that perhaps he's being a little blind himself here. He says, he knew not the shape of anything, nor any one thing from another, however different in shape or magnitude. But upon being told what things were, whose form he knew before from feeling, he would carefully observe that he might know them again. But having too many objects, and so that sounds like, after he's learned to feel what they look like, he'll know what they look like. But then he hedges things a little bit. He says, but having too many objects to learn at once, he forgot many of them. It's a new world from him. Everything looks different to him, and so he's got to remember a lot of stuff. And as he said, at first he learned to know and again forgot 1,000 things in a day. One particular only, though it may appear trifling, I will relate. Having often forgot which was the cat and which the dog, he was ashamed to ask. But catching the cat, which he knew by feeling, he was observed to look at her steadfastly, pardon me, those F's and S's, and then, setting her down, said, "So, puss, I shall know you another time." He was very much surprised that etc. So, in fact, it sounds like long after he had his vision, he still couldn't tell from looking at it which was a cat and which was a dog. And Cheselden attributed that failure to recognize objects to having a crummy memory. Well, he just didn't remember what they looked like, etc. And so that might strain belief. Right? In other words, Cheselden, like most researchers, saw what he expected to see, got the results he was expecting to get, and interpreted them the way he wanted to. So latter-day cases are not nearly as optimistic and make it pretty clear that Cheselden was mistaken, that what he attributed to faulty memory was, in fact, an inability to use vision to distinguish objects. So, in fact, these days, if people have the cataracts removed late in life, they are very poor at recognizing objects, and especially poor at looking at faces. So here's a famous example, a fellow named Michael May, who was born with sight. But when he's 3 years old, there was a chemical explosion, and he lost one eye, and the other eye was so clouded that he couldn't see form vision, right? So it's sort of like having a cataract. And they tried doing a corneal transplant when he was little, but it didn't work. So he must be an amazing person, because despite the fact that he was blind, he never let that stop him. He loved riding bicycles. And so he would ride his, bicycle around the neighborhood, and his parents didn't make him stop until he wrecked first his brother's bike and then his sister's bike. And they said, well, that's enough of that. He learned to play a pretty good game of ping pong, but only in his basement, where he learned from the sound of the ball to guess where it was going to be, right? He grew up, he wanted to learn how to ski, downhill skiing. So he learns how to ski, and he falls in love with a ski instructor, and they have a family. Oh, oops. Oh, none of the food lost, I hope. And et cetera. Then, as an adult, after decades of not being able to see, technology allowed him to restore form vision in the remaining eye. And he comes up out of the surgery, and his wife's there, and he can tell you that that's his wife's, that she has blonde hair, which he knew from being told. And he can tell you other things about her face, but he doesn't actually recognize her. It's not until she talks that he recognizes her. And so you say, oh, well, but of course he had to, as Cheselden and Molyneaux and Locke predicted, he had to learn what she looks like in order to know what that looked like, to store it in memory, right? But decades later, he's still not able to recognize anyone by face. And they gave him a test where they used Lego objects like this. And when newly sighted individuals can only touch them, it turns out, even if you give them lots of experience touching them and seeing them with their new sight and touching them, they never learn how to distinguish them by sight. And for Michael May in particular, so immediately after his surgery, he was very poor at detecting objects. In fact, his performance was no better than chance alone. But that's okay. He needs time to learn what things look like. And yet, 10 years later, he still couldn't tell what things looked like just by looking at them alone. So Molyneaux and Locke were more correct than they knew. Not only will the individual not be able to distinguish objects by sight to begin with, he will never be able to make use of his sight at all. Not what they were expecting. And we can understand what happens to these individuals. Oh, by the way, and for this reason, nowadays, when babies are born with cataracts, they try to correct it as soon as they can, as soon as the surgery is safe, given anesthesia and stuff like that. And babies in which the cataract surgery is delayed, say a year, when they grow up, they make use of their vision, but they are not as good at distinguishing faces as control individuals. Just that period when babies come out and they're so fascinated with faces and looking at them all the time. If babies don't have that period where they're studying lots of faces around them, when they grow up, they're not very good at recognizing faces. And we can understand these phenomena based on, good. How?
Is it just for like faces of objects?
In this case, it depends on how early the surgery is done and other things like that. In general, the highest threshold is recognizing human faces.
Yeah.
But in those cases, I think I don't remember that they had any significant deficit in recognizing objects, but for them, it was specifically faces. So you have to have visual experience if you want to have a visual system that can work. And the highest challenge for a visual system is distinguishing human faces. We are unbelievably good at it. Right? And you can imagine how natural selection wants us to be unbelievably good at it. And it could be that the reason natural selection arranges babies to find faces so fascinating is because it's really important for them to get to know them and get to distinguish what different faces look like. So there's a series of neuroscience experiments that began in the 60s that can explain to us what's happening in these cases. And it is one that is important in the history of neuroscience and important to you as a neuroscientist. You cannot call yourself a neuroscientist unless you know about Sperry's experiments with frogs. And you cannot call yourself a neuroscientist unless you understand Hubel and Wiesel's experiments looking at the developing kitten visual system. Okay? And so if you haven't heard it yet, here you're going to, and after this, you all get a membership card, neuroscientist. It says so right there. To understand this research, you need to know something that was way back here, which is you need to know something about the ontogeny of synapse formation and number in cats. So here are cats, and we have lots of different things being measured on this same axis. One is brain weight. From 50 days of age to adulthood, the cat brain grows quite a bit, right? From a few grams to maybe 30 grams. And most of that change in brain weight, by the way, is due to the fact that neurons are getting bigger. Not more neurons. There aren't that many new neurons coming along. Neurogenesis continues throughout life, but it's pretty slow at this rate. On the other hand, we can look at the volume of individual neurons, and you can see that they're responsible for most of that weight. The crucial thing is how many synapses are there. And as you see, cats, after their eyes open, about a week after birth, 10 days, I forget, we can keep track of the number of synapses per neuron. And this, in particular, is in visual cortex in cats. If I showed you, numbers from other areas of cortex, the graphs look much the same. And you can see that during this period, from 20 days of age to, say, 50 days of age, there's a lot of synapses coming, and after that, the number of synapses goes down. And it turns out that this phase of making synapses and losing synapses is being controlled by visual experience in cats. And so let's talk about how we know that. So in these experiments, first of all, I have to deal with the fact that these are experiments of people doing manipulations in kittens. and we all love kittens, and they're cute as can be, and there's just no way around it, right? That's what they were doing. And most of their experiments involved depriving kittens of vision. Sometimes they would do surgery and suture an eyelid close, so there would still be light coming through the eyelid, so that the eye would detect whether it's light or dark, but there'd be no form vision being focused on the retina. Everyone see that? In other experiments, instead of suturing the eyes closed, they put a frosted contact lens on the eye, which was probably more uncomfortable for the kitten, but it accomplished the same things that we're talking about here. So they would do manipulations like this, and then they would let the kittens grow up, and then they would kill the kittens. They would anesthetize them, expose the visual cortex and the occipital cortex, and stick electrodes into the brain and record the electrical activity of neurons there. And following in the footsteps of people who had done similar things in the tectum of frogs, they learned that they could anesthetize the animal and put a screen in front of it. And by moving light around on the screen, sometimes they could make the neuron they were listening to in visual cortex fire. Everyone get that? And the question is, for each neuron, what's the best stimulus to make that particular neuron send lots of action potentials, right? So they're mapping the visual field. These experiments are terminal. So after the experiment, they would do the experiment, keep the animal alive as long as they could, and when it was over, they'd kill the animal and take out the brain and detect exactly where they were recording. Anyone eager to do that kind of science? Good. That's good. Any questions about it? Because there's no getting around it. That's what they did. Other people would do similar experiments in monkeys, but the famous research got done first in cats. You're okay? Yeah, Erica.
So why is that
Say it again.
reason why they chose hen?
Why did they choose cats? Yes, number one, they're a lot cheaper than monkeys. And this was in the day when it was easy to get plenty of cat subjects because it's probably still true today. Animal shelters in the United States, they kill like 10 million cats and dogs a year, where they find them, nobody ever comes to adopt them, and they've got limited resources. They can't keep them all, and they kill them. Notice that I just want to point it out to you. Every one of these animals is an abandoned pet. Every one of them. The only reason they had to be killed is because somebody made them and then didn't take care of them. And I'm sorry, we're good at making cats and dogs in the United States. We're not particularly good at taking care of them. And therefore, in those days, you could get cats from a shelter that was otherwise going to kill them. You didn't, you had to do it through official channels, and the campus veterinarian had to inspect them and all that stuff. But they were relatively cheap to get. There's no way to get monkeys like that, right? And other questions about that. Also, it's easier to keep cats in a lab than dogs, right? They get bigger. It's a great question. And I guess I don't know all the answers. I know they wanted a mammal, because all this stuff was pretty well worked out on amphibians already. Okay, Hubel and Wiesel, they were at Harvard. They won the Nobel Prize for this, along with Sperry and later. And you can judge for yourself whether it was worth it. Okay. When they looked at normal adult cats, they noticed something interesting. They noticed that in layer 4 of visual cortex, every cell was monocular. It was responding either to the left eye or the right eye. It would be several years later when Simon LeVay, working with them, pointed out to them that segregation of inputs to layer 4 into monocular bands, that it was visual activity, was activity dependent, but they didn't know that yet. And they expected that physiology, that anatomy had been pretty well worked out. But they noticed that if they were anywhere else in visual cortex other than layer 4, all the neurons there, most of them were binocular. So they would find a few cells that would respond only to light in the opposite eye, the eye on the opposite side of the animal. And so they came up with a 7-point scale. They rated a neuron that they could only get to fire by shining in the opposite eye. They rated it a 1. Other cells would only respond to light in the same side, and they called that a 7. And the most common kind of cell was one where it was equally driven by both the left eye and the right eye. So these are binocular to various extent, and these two are monocular. Now, when they found a cell that responded to light in both eyes, these were not random parts of the two retinas, right? When a cell was binocular, that's because a given point in visual space was stimulating homologous regions of the two retinas, and those are the retinal cells who are synapsing eventually on this particular neuron. Everyone get that? In other words, each cell they recorded from was responding to a part of visual space, and it would respond to that visual space whether the light got there in the left eye or the right eye, just as you'd expect. How's that so far? And again, this is outside layer 4. So this is what the pattern looked like in normal cats, and you'll hear these talked about as ocular dominance columns. I don't know why. How do we get rid of that? Well, I don't remember what it says. Oh, I can tell you. What does it say down there? Probably nothing important. Sensitive period. These manipulations affect ocular dominance. Oh, only if done early in life. It turns out to be important, but I haven't told you what the manipulations do first. What they noticed is if they did monocular deprivation, and if they did it for a medium amount of time, let the animals grow up, take out the sutures, let the animals grow up, do the experiment, kill the animal, and do the experiment. What they saw was in these animals, almost every cell was monocular, outside layer 4, and they were all responding to the eye that had been open. And there were very few cells that would respond to light in the other eye, the one that had been deprived. And what they found eventually is if they did the monocular deprivation long enough, the animals grew up to be blind in that eye. And the reason they were blind in the deprived eye is because the connections to visual cortex had all withered away. They weren't there. In that eye, they're like Molyneaux's problem or Cheselden's patient, i.e., they'd done without form vision in that eye for so long that they were functionally blind there and could never make use of it.
So,
Interestingly, if you deprive an adult cat of vision in one eye for that same length of time, and then open up the eye again, they're totally unharmed. They couldn't find any evidence that this made a difference. So these effects on ocular dominance columns only happened if you did them early in life. And the earlier in life you did them, although you don't bother starting until their eyes open, about 10 days or so. The earlier in life you do them, the manipulations, and the longer they last, the more profoundly they affected circuitry from the eye to the visual cortex. And they were measuring that circuitry through physiology. Everyone have that so far? Interestingly, if they sutured both eyes for a medium amount of time, it didn't have much of an effect. That is, when they opened their eyes as adults and did the experiment, they saw that most of the cells were still binocular. For all the world as if, with both eyes silenced, neither eye could outcompete the other and take over visual cortex. So in that respect, it turned out depriving both eyes of vision wasn't as deleterious as depriving one eye of vision. How's that? And these experiments, even more than those, let's see, I should give you a little more historical text. Removing cataracts in babies, human babies, that the importance of doing that early wasn't known yet back in 1962 and 63 when they were doing these experiments. Because everyone thought, oh, you can wait till they grow up, and nobody bothered to see how good the form vision was in those babies when they grew up. But these experiments changed medical practice in lots of ways. One was that, well, first of all, the interpretation was that the visual system will not work unless there's visual experience during a sensitive period in development. And that if you wait until after that sensitive period of development and provide visual experience, it's too late. The eye will never be able to make sense of the information, and the person will be functionally blind. And so that was a big impetus for people to go back and look at the effects of removing cataracts in babies, and that's when they realized there was a deficit if you didn't do it early. And that's when medical practice changed. If you want the child to have the best form vision, you get rid of the cataracts as soon as is safely possible. I'm doing this deliberately because if you think the kitten experiments were cruel, and I don't think you can get around that they were. I mean, they tried not to make the animals uncomfortable. It was cruel. At least there was some benefit from it. And the number of children that have benefited far outweighs all the cats they ever used, right? So you got to ask yourself, is it worth doing that to kittens if it's going to provide vision to 10 times more people? And there's no scientific answer to that question, of course. Questions about that? And so why can't Michael May make sense of what he sees? Why couldn't Cheselden's teenage patient ever seem to quite remember what cats and dogs looked like? Because with the absence of visual experience early in life, they simply didn't have the circuitry in the brain to distinguish what objects look like. They were, in many ways, blind. And the very fact that they could tell that there's something in front of them is probably due to the fact that they were getting some form vision past the cataracts. Michael May, the fact that he is able to use his vision at all, is probably due to the fact that he could see until he's 3 years old. So, yeah. Although he says that when he's skiing, it's too confusing to have his eyes open. He still skis with his eyes closed. That's safer for him. There's another aspect of the Hubel and Wiesel. I guess I don't have that name anywhere, but it is certainly in the book. Hubel, Hubel, and Wiesel, Wiesel, at Harvard. Another aspect of their work changed medical practice too, and that was an experiment they did where instead of suturing one eye closed to the other, they induced a strabismus, where they caused the cat to have an eye that was either went wide or the eyes came together. They did this by doing a surgery where they would cut one of the extraocular muscles so the eye would drift in the wrong direction. And these were very interesting experiments. First of all, to understand the outcome, you need to understand that when one eye is largely deviated from another, that an object in visual space is no longer reaching homologous portions of the two retinas, right? Because one eye is so misdirected that now it's two more or less random parts of the two different retinas that are being stimulated. They are no longer in homologous regions. Everyone understand what I mean by homologous there? And so what they found is when they induced strabismus, that there was a virtual loss of any binocular cells. No matter where they were in visual cortex, each individual neuron was either listening to one eye or the other eye, and very few were listening to both eyes, which is the normal case. And they didn't do the behavioral analysis, but they concluded that probably these cats had very poor depth perception. right? Because they were no longer able to see the difference in input to the two eyes and make sense of it, etc. Well, it turns out that strabismus happens in people too. And one of the things that people learned is when children are born with a strabismus, that if you don't intervene at all, eventually they come to suppress the information in one eye. So that they will end up preferring either the right eye or the left eye. And if you close that eye, they can't make sense of what comes in the other eye. So they have, at some level, they have suppressed that information. Everyone get that? And sometimes that happens in children who aren't having a strabismus, where for whatever reason, it turns out with an examination, and this is why eye exams in schools always test both eyes. When you do an eye examination, it turns out that they can't tell you what they're reading. They can't tell you what the chart looks like in one eye or the other. And they look at the eyes and they seem to be lined up, and they'll look to make sure that there's no cataracts, make sure that there's, that the image of the visual world is being projected crisply on the retina in both eyes. And it turns out that there's nothing organically wrong with the eye. And this is sometimes referred to as amblyopia, or you might hear referred to as lazy eye, as though one eye is too lazy for the individual to use it. And there's a very effective treatment for this, which is you make the child wear a patch over the good eye until they learn to make sense of the information in the other eye. And so if you've seen children with a patch over the eye, and if there isn't some injury going on, this is what's going on. And, if you do it for the right amount, eventually they're able to make sense of information in both eyes. Likewise, if a child has a strabismus and you patch the good eye, they can eventually learn to make sense of the information in the other eye. But better yet would be surgery to correct the strabismus. Until Hubel and Wiesel did their experiments, the idea when a child was born with strabismus was that it was purely a cosmetic problem. And you didn't bother trying to surgically correct it, if you ever did, until they were old enough that the surgery and the anesthesia was safer. Everyone see the rationale for that? But it turns out that if you wait too late to correct the strabismus, that they're never very good at detecting at depth perception. So here's a chart where they're keeping track of individuals who had strabismus corrected at different ages. And notice that in children where the strabismus is corrected by 4 years of age, they have good depth perception. If you wait much longer than that, depth perception is not nearly as good. This individual seems to be an outlier, but these individuals, it's much less. And if you wait until they're grown up to correct the strabismus, it's too late. They're never going to have good depth perception. So, because of data like this, now medical practice is, again, you correct the strabismus as early as possible, not because of any cosmetic concern, but because you want the child to have full vision. How's that? The conclusion of all this is that the way natural selection caused your visual system, back then to the case of strabismus. Well, if this means that those binocular inputs are lost, how come back here, right? How come back here both inputs weren't lost? Well, number one, neither eye was sending out information, but until the strabismus happens, probably just like in frogs, probably the initial projection to the brain is probably initially topographic and is roughly correct. And that's why when the eyes open up, any neuron in the cortex that's responding to a light in space is responding to light from homologous regions of the two retinas. See, that wouldn't work unless from the very beginning, there's roughly correct projection from the eye to the brain. But that's not enough for them to actually make sense of the information. And if you want them to make sense of the information, they're going to have visual experience during the sensitive period. Okay, now questions about that. So here's this idea that a sensory system, in order for it to actually work properly, natural selection, you could imagine natural selection might have invested enough genes in some complicated mechanism to make sure as soon as the eyes open up, the vision is super crisp, right? But, and I can't tell you why natural selection didn't do that. I can just tell you that I can imagine that's a pretty hard task to encode in genes alone. But natural selection hit upon a better solution, which is get things in the roughly correct position, as in frogs, as here, and then let the world teach the animal and the brain, what things look like, and learn to analyze the visual world that's around them. And what we're going to see is this is probably true for every sensory system. I do have to, though, show you one thing where, I love this illusion, and so you already know it's illusion, and I do wonder why it works. So most of you probably think that the reversed numbers are slanted more, and the reversed letters are slanted more than the ones up there. But they're not. I just took these and flipped them over. They're not more slanted. Why do you see them as more slanted if they're in one orientation than another? I think it might have something to do with the fact that you've all had lots of visual experience with things that look like this and things that look like that. And what it suggests is that when you're reading italic print, for example, without even being aware of it, you undo the slant a little bit to make it easier to read. And so I'm eager for someone to do an experiment with people who have never seen italic print. Let's say it's an easy experiment. Raise them in an environment, make sure you have everything they read. Okay, I can see some problems. And to see whether they'd be subject to this same illusion. Questions about any of that? I.e., it might be that this illusion is a result of your extensive visual experience reading and being around clocks and stuff like that. Okay, definitely time for a code. Anyone disagree? Okay. What we're going to see next is this general principle, that is that sensory experience early in life is not just good for, but is absolutely crucial for sensory systems to develop such that an individual can actually use them, to take care of life, to stay alive, reproduce and stuff like that. So one example I want to talk about is in the olfactory system. Remember, this is the place where olfactory sensory neurons are continually generated throughout life. And the olfactory sensory neurons, let's see, first the air comes through the nasal passages and reaches the olfactory epithelium, way back up there. And in the olfactory epithelium, underneath that slime, there are a layer of cells that include olfactory sensory neurons. And they stick their dendrites into the slime, and when a chemical odorant lands on the dendrite, they recognize some odorants and not others, and they send an action potential through the cribriform plate at the base of the skull to synapse in the olfactory bulb. Within the olfactory bulb, they're going to, each cell is going to synapse in a particular sphere of cells that are all kind of talking to each other. That's known as a glomerulus. Hope I have that there. Yeah, glomerulus. Hard to say. And it turns out that for the olfactory sensory neurons to detect the odorant, they have to make a specialized transmembrane protein that they insert into the dendrites. And the outer part of the protein has a receptor that recognizes the odorant. Right? Everyone got that? Makes sense? And it turns out that each sensory neuron makes one and only one olfactory receptor protein. Okay? And therefore are specialized to detect a particular odor. What's fascinating, and I haven't heard any explanation of it yet, but kind of boggles the mind, is that those sensory neurons that are expressing a particular olfactory receptor protein, if they're all expressing the same olfactory receptor protein, they will eventually synapse in only one of two glomeruli in the olfactory bulb. So it's like each olfactory receptor protein detects a particular odorant, and that odorant will be reported to two and only two glomeruli in the olfactory bulb. Just the question of how that got arranged in the first place is pretty fascinating, and nobody knows the answer yet, but that's just the fact. What I want to talk about is the fact that early in the, and here you can take a look at that. Nope, wrong direction. And it turns out that one of the reasons rats and mice and cats and dogs have so much better smell than you do is that many of the olfactory receptor proteins that our common ancestor had have in humans become dysfunctional. They've accumulated mutations and they don't work anymore. So let's see if I got the numbers there. I guess not. So it turns out that rodents, dogs, etc., they have, tens of thousands of different olfactory receptor proteins, each with its own separate gene. And so they can detect many more different odorants than you can. That's one reason why they have better smell than you do. And you might ask yourself, well, how come those genes degraded in humans? Presumably because they weren't very important for making more humans, right? And so they accumulated mutations, became dysfunctional, and nobody's missing them. Another reason why animals have better olfaction than you do is because they have a lot more olfactory sensory neurons. So here, showing a picture of the size of the olfactory lobe in rats. Look how big it is compared to the rest of the brain, because that is to scale. And compare that with humans, where the size of the olfactory bulb is so much smaller relative to total brain size. In fact, the human olfactory bulb in actual size is smaller than a rat olfactory bulb. So that's why, they're devoting a lot more sensory processing to odors than you are. And that's a third reason why they have better smell than you do. Okay? And it turns out that those olfactory receptor proteins, each olfactory sensory neuron is expressing only one. Those olfactory receptor proteins, it turns out they derive from 4 different families of proteins. That is, there's 4 different groups who all have more similarity to one another, and presumably because they, evolved from a common ancestor gene. And here, one of my favorite photomicrographs, here people have used pseudocolor to keep track of where all the olfactory receptor neurons are for those who are expressing the green kind of protein, the yellow kind of protein, the blue kind of protein, and the white kind of protein. And so here's this lovely topographic map of the olfactory world. So when I show you a topographic map of the retina onto the cortex, you know what they represent. What does this map represent? The topographic map in visual system is the visual world. What are these 4 dimensions of odor that are being displayed here? Odors that are significantly different from each other on some sort of continuum, I suppose. Anyone know what it is? You're in good company. Nobody else does either. And in fact, to this day, one of the biggest stumbling blocks in olfactory research is people are having a hard time finding out which odorant is detected by which protein. It's a bit of a mystery. But presumably there's a spectrum, a spectrum of odors that's on some scale we don't have a name for. You know, in what way is cinnamon kind of like nutmeg? Okay, I think those are similar. How about cinnamon and garlic? How do they differ from each other? I don't know. I don't have any words for that. Okay. Now to get back to development. Oh, and here it just says what I just told you, that blah, blah. It turns out that when you look in young rodents, that originally, in fact, each glomerulus is getting input from several different kinds of receptor proteins. So here, it's a complicated experiment. So the nostrils open, and here we're keeping track of the few olfactory sensory neurons that can't respond to odors that survive. So this is in the occluded nostril. And here are the distribution of olfactory sensory neurons that detect a given odor, i.e. none, they're gone. But on the other side, where the nose isn't clogged up, that olfactory bulb is getting a lot more unresponsive olfactory sensory neurons that are surviving and project to the bulb. So in these experiments, they deliberately disabled one of the olfactory receptor proteins genetically. And when they did that, the ones that weren't effective, pardon me, I got it. When they did that, the ones that weren't effective, when they were competing with other odors, they lost their input to the olfactory bulb. But this is the side that's occluded. Sorry. On this side, where the defective olfactory receptor protein wasn't competing with any other receptors or receptor proteins or receptor sensory neurons, their projections to the olfactory bulb persisted. And so here you can have another view of it, where here's the side where the defective ORPs, the defective OSNs, lost out. They no longer project to the olfactory bulb, presumably because they got pushed out by the functional olfactory receptor proteins. Whereas if we, just like when we keep both eyes closed and there's no competition between the two eyes, if there's no odors coming in to that olfactory epithelium, then the defective olfactory receptor protein and its sensory neurons, they continue to project to the olfactory bulb. In other words, it's pretty clear that olfactory experience, and it turns out you have to do these experiments early in life for it to happen, that olfactory experience is also guiding inputs in the olfactory system. Questions about that? Good, Erica?
And there are also some like actually.
Are there also behavioral deficits in these animals? Yes, but I mean, what we can say is that the olfactory, if one nares, one nostril, if it's closed for very long, they'll never be very good at detecting odors in that particular nostril. But in terms of, I mean, it would be great if we knew, and the reason they did this experiment is they thought, well, if we disable one particular olfactory receptor protein, maybe we can test the animal on lots of different odors and find out which one's missing. They never could find out. And these experiments have been done several times. I haven't kept up a lot. There are a few receptor proteins where they're pretty sure that they do detect a particular odor, but there's tens of thousands of them, and most of them, nobody can figure out how to tell what. And I should point out to you that even though you have, I forget, maybe 5,000 olfactory receptor proteins, we know from psychophysics that you can detect many more thousands of different odors than that, right? So the way the olfactory system works, it's not just that there's only one protein for each odor, right? I mean, each protein is specific to an odor, but the way you make sense of it is there's some sort of coding where particular patterns of stimulation indicate one odor and a different pattern of stimulation would represent another. So in other words, you can distinguish more odors than there are functional olfactory receptor proteins in the human genome.
Would that also suggest that there's combination of them?
It must be that one way we distinguish odors is by the combination of inputs getting stimulated. Combination of channels, if you will, of receptor proteins. No, I shouldn't say channels because I'm not talking about ion channels, but you get the idea. Good, yeah.
So that said that this was performed in like young rats.
In this experiment, I think mice, but yeah, in this experiment, yeah.
So if they were adults, then it wouldn't have the same effect?
Oh, well, first of all, they couldn't do it exactly this way because the defective olfactory, but yes, it's true that if you occlude the nostril later in life and then undo it, they can still detect odors in that nostril. So to have the effect of making them unable to use olfactory information on one side, you have to deprive them of that stimulation at a particular sensitive period. All pretty similar to what was seen in vision. And I'm going to pick one more modality that suggests that maybe this is true for all of the visual systems, all the sensory systems. So in this case, we're going to move from olfaction to tactile information, touch. And I want to hark back to a particular homology with the visual system that might be helpful for you. Remember we said that when having early visual experience, an object in visual space is stimulating homologous regions of the retina. And when we talked about that competition between the two eyes for layer 4, we said an important way for that to work is that on the retina, neighboring retinal ganglion cells are more likely to fire at the same time. Put another way, choose any retinal ganglion cell, and the other retinal ganglion cells that are close to it are more likely to fire at the same time than a randomly chosen one, right? Why? Because objects in visual space stimulate neighboring regions of the retina, right? And that means that an object in visual space stimulating neighboring retinal ganglion cells, they'll fire at about the same time, and they're more likely, therefore, to use Hebbian's synaptic strength to take over a particular part of the tectum in frogs, or a particular part of the lateral genicular nucleus, etc. Got that? You might not have thought about it before, but that same principle is true for the tactile system, for touch. In real life, you almost never receive a touch that is a tiny little pinpoint, like the Von Frey hairs you might have seen people use when they're doing psychophysics. That's not what real life is like. In real life, when you have touch, right, when you are, when someone hits you with a stick, they don't hit one receptor in your skin, they hit a bunch of neighboring receptors in your skin. Or when you're feeling an object, you're not just feeling with one receptor, you're feeling it with a bunch of receptors who are all close to each other. Everyone get that? And in fact, here is a topographic projection of touch. And this is specific in rodents who have whiskers, those little vibrissae that stick out. If you've ever had a pet, you may have noticed that they're not distributed randomly. They are in a very tightly packed, orderly array of whiskers. And every rat and mouse has the same number in the same order. And if you look in somatosensory cortex in these animals, there's a huge chunk of somatosensory cortex that is not detecting touch receptors anywhere except at the whiskers. Each whisker has at the bottom of it touch receptors that detect when the whisker is being deflected. And people were kind of surprised when they took particular sections parallel to the surface of somatosensory cortex. They saw these barrels that in ordinary Nissl staining looked darker, And it turned out the number of barrels was exactly the same as the number of whiskers. And so, in fact, in somatosensory cortex, these barrels are represented in regions. And if you do recordings, you find that the cells in this region, in this barrel, respond to a particular whisker and no other. And it turns out that this projection of the whiskers onto the somatosensory cortex is also dependent on early sensory experience. Because if in young animals you lesion a line of whiskers, when you grow up, there's not like a missing part of the, there's not a part of the cortex that's getting nothing. Rather, the other whiskers expand their fields and am now sending information to a larger part of somatosensory cortex, so that every somatosensory cortex neuron is responding in this area, is responding to one whisker or another. And if you get rid of a row or if you get rid of a column, it's the same. And if you get rid of all of the whiskers, then the barrels never form in the first place. So it's pretty clear that the whiskers have to have sensory experience in order for them to have any chance to talk to somatosensory cortex. Questions about that? Yeah.
Is there any order to like how the barrel grows within the brain?
Tell me more. I mean, they as it happens, they are arrayed on the somatosensory cortex in the same spatial relationship between the whiskers that you see on the muzzle. Is that?
Yeah, but like when they grow, is it every single one's growing or just the one, like the whiskers that correspond?
Oh, you mean before they're functional, when they're starting to grow?
Yeah, when it's developing.
Yeah, I don't know enough about the whiskers. I mean, the part of the muzzle that's going to make the whisker is already there at birth, but of course there's no whisker, it hasn't been made yet. So I don't know. I actually don't know about that process. Something must lay them out in an orderly array.
Oh no, I'm asking in the
somatosensory cortex. Okay.
To the whiskers that correspond to, that are close, the whiskers that are gone, that are cut off. Do they grow more than other whiskers?
Do the whiskers themselves get bigger? No. But the portion of somatosensory cortex they innervate absolutely gets bigger.
And is it?
So here's the control. In this region. Here's what the control. They took out, right, this area. It's this vision. They took out. Let me see. Here they took out the middle. This one here, and the ones on either side expanded their representation in somatosensory cortex to take that over.
Okay, so it is the ones closer.
Yes, absolutely. I see. Yeah, and I don't think there's any evidence that all the barrels got a little bit bigger. You can imagine it was like that, but it was the neighboring barrels that got significantly and obviously larger.
Is that due to like more touch? Do they get more touch? You could be the other ones they're missing.
I don't know. I don't know. But presumably the reason they took over this empty, this newly vacated lot is because they were nearby. And so you get the impression that this nice orderly array of the barrels in the cortex, that it happens because the various whiskers are competing with one another for space on the somatosensory cortex. And if they all equally compete, they all get a pretty much same size barrel. And there were other experiments like where you might glue several whiskers together, right? And therefore they all are fired at the same time. And then instead of making 3 barrels, they make one big barrel, right? As if, okay, they're all competing there. This sort of competition in somatosensory cortex also happens in humans, and it's pretty clear that it happens throughout life.
Yeah.
So in a famous set of experiments by Merzenich et al. In the 90s, maybe, they were doing experiments with monkeys. And it turns out the projection of the fingers of the hand on the somatosensory cortex is somewhat more complicated than you might think. And I'm not going to, but here we are keeping track of the 5th digit and the thumb, right? And they have different portions of somatosensory cortex where they are sending their input. And we keep track of this in monkeys by recording the activity of neurons in this various portions of somatosensory cortex, and then touching the various fingers and seeing which fingers they respond to. So that's how maps like this got made. Okay. And so this is the normal pattern of information from the various digits reaching somatosensory cortex on the opposite side of the animal, of course. And in one experiment, they surgically removed a digit, and they did this in an adult monkey. Okay. So, and then after they waited a while, they did the recording, and what they saw was that the neighboring digits, they expanded their representation on somatosensory cortex to take over the space in between. In other words, when they did the mapping, it's not like there was a blank region here where the third digit used to provide information. Rather, the other two regions took over. And that too kind of sounds like it could be a Hebbian synapse sort of thing, right? As long as all the neighboring receptors on the digit are firing, often firing at about the same time, they're going to predominate, and therefore they're going to be able to have their synapses effectively fire the postsynaptic cell, and so those synapses stay. If they're, but if the receptors are gone, then they can't compete, and now it's the neighboring receptors that are going to be competing on the neighboring digits, and they're going to take over that space. And they didn't test this here, but you have to wonder if the remaining digits, or at least the neighboring digits, if the animals, now that they were using more neural space to analyze that information, maybe they're better at analyzing, at detecting things in those fingers. But they didn't do that. They did a cooler experiment, which is they gave the monkeys an experiment, a task, where the animal had to stick its hand into a box where it couldn't see, and there was a disc rotating. And whenever the animal felt a particular change in the disc, they were supposed to push a lever to get apple juice. And monkeys will do anything to get apple juice. They love apple juice, right? And by the way, these experiments are very difficult. When you see people in the literature where they're training monkeys to do anything, first of all, the reward's always apple juice. That's fine. But in fact, it takes a long time to train a monkey to do these things. The first thing is you have to train them to want to eagerly jump in the chair, right? And be restrained so they can do the task. And if they know they're going to have a chance to earn apple juice, they jump in the chair, no problem, which is good because there's no way you could possibly force them into that chair, right? But it also means that every time they're doing these experiments, the experimenters are working with these monkeys for a long time. Anyway, so they have this monkey where they choose two digits and say, these are the two digits that you're going to be able to detect the signal to let you know when to get apple juice. And lo and behold, when they mapped somatosensory cortex later, they saw that the region that was analyzing information from the two important digits had expanded. Somehow, the monkey, by paying attention to those two particular digits, paying more attention to those two digits than the other, they had managed to expand their input to somatosensory cortex, and that probably made them better at detecting the signal with those two digits. So far, so good. And I think the surprise for everyone is that these effects of sensory experience in the tactile system seem to be going on lifelong, because all these experiments were done in adult monkeys, no sensitive period. And so you get this idea that, our somatosensory input, our tactile input to somatosensory cortex is a matter of neighboring receptors beating up on and coming to dominate particular neurons in cortex. And as long as they're active and the neighboring cells tend to fire about the same time, they'll continue to have representation in the somatosensory cortex. But if they go quiet for some reason, then the neighboring cells will be the ones that are firing more often, and it'll be their Hebbian synapses that get stronger. And then too, you think, oh, in the course of evolution, maybe when natural selection hit upon these maps of somatosensory of the body onto somatosensory cortex, maybe it was a good idea that natural selection hit upon these ways of getting the map there, because, in the course of evolution, animals' bodies in a given species, lots of changes are taking place. How much harder would it be is if natural selection causes a body modification to happen, the arm gets longer, the fingers get larger, something like that, to also then arrange at the same time for the amount of brain space devoted to it to get larger. That'd be a significant hurdle. On the other hand, if body representation in the somatosensory cortex is simply a reflection of what the body is, if they're all competing, well, then it always works out. No matter how weird a body you have, you've got a topographic mapping of the body onto somatosensory cortex. Haven't lost you yet? And it's clear that this happens in people too. I'm going to skip over this for a little bit. It's just that rat dams, before they're going to have pups, they do this behavior where they start cleaning their nipples, getting ready for the pups are going to come. And it turns out you can show that when they've been doing that and when the pups have been nursing, there's an expansion of their somatosensory cortex in the nipple area. And that too happens in adults. And these things are probably going on in humans too, because the map of somatosensory, and here, these experiments we can do with non-invasive imaging, functional MRI. And normally, somatosensory cortex is such, there's this weird bit that the hand representation is wedged between the face and the upper arm. So it's like you're built with your hand in between the two. That's how it looks on somatosensory cortex. Right? And it turns out that when you look at people who have lost a hand that's been amputated, it's not that there's a blank spot there in somatosensory cortex where the hand representation used to be. Rather, the arm and face expand to fill up that space. And in something that could only happen recently, people found that in one man who had lost an arm as a teenager, and then something like 30 years later, they were able to transplant a new hand, somebody else's obviously, onto him. And he eventually was able to get sensation there. And when they mapped his somatosensory cortex, that region that used to be listening to the face and the upper arm, now it was responding to the hand. So once they were online, they were able to compete for space again, and he was able, and therefore they appeared in somatosensory cortex, which, by the way, is the only reason he could make sense of that information in the first place. That's how he knew that his hand was back. Right? Oh. Questions about that? Okay. I'm going to go a bit further. And so this is just to remind you that after birth, there's a big proliferation of synapses in the cortex. And we talked about this already. I've shown you this figure, right, already? And when we get back, we'll talk about the fact that oligodendrocyte myelination in the brain is also responding to experience, okay? And when we finish talking about that, we'll have our Kahoot quiz and we'll start chapter 10 on Thursday, okay? But, I mean, you can print out the notes for chapter 10, but don't read them. Just bring them with you. Okay.
One, so